So as I mentioned, I don't have anything planned or prepared. So I'm here to talk about whatever you guys want to talk about. And probably largely we'll try to do word work, but sometimes we'll do slides. What would you like? Oh, yeah. Yeah, so the idea is, this is just basically, I call it the pathway of information flow because it's, it's part of it is describing organelles, part of it's describing processes and such, but this is the process by which we make a protein and eject it. So if we were making a peptide-based neurotransmitter, for example, or a receptor that would be found on the membrane surface, the idea is you're starting with a double-stranded DNA molecule, and that's found within the nucleus. Is what we're doing, right? Yeah, and then what we end up doing is we transcribe it. So there's a RNA polymerase that comes down and it binds to the DNA and unwinds it, and from that we create a messenger RNA molecule, which is your primary transcript. I think I'm using those terms right. You guys probably know that are in cell biology. And then from there, we will splice, because the messenger RNA transcript has lots of introns, and we're interested in the exons. And so as a result, we'll cleave out all the introns, leave the exons, and so we end up with a fragment of messenger RNA that is much, much shorter than it had been before. And at this point, this thing is ready to exit the nucleus via a nuclear pore, and then come out here into the cytoplasm. And at that point then, it'll bind to and interact with the ribosome. The ribosome could just be a free-floating ribosome, but oftentimes, especially if we're interested in something that's going to go to the membrane or be released from the cell, usually in those cases, the ribosome will couple with the messenger RNA molecule, but then, so this is the ribosome, but then after it couples with that, it'll then bind to a docking site on the endoplasmic reticulum. It'll be the rough endoplasmic reticulum, because the thing that makes rough endoplasmic reticulum look rough is there's ribosomes docked all over it. At that point, you'll have translation occur, and you'll get a protein, or at least a peptide, produced here. So that process is called translation, where we're going from, it's translation because we're going from a nucleic acid language to an amino acid language. And so we're sort of going into a new character, using new characters than we were before. So it's very much like a new language. Whereas transcribing, we're really just kind of going from DNA to RNA, which more or less uses the same language. So you're just copying, transcribing. Okay, so at that point, we've made a peptide. The peptide will do there'll be some folding that will occur. There'll be other kinds of things, we call them post-translational modifications. Which just means what it says. Any modifications, any modifications that occur after translation are post-translational modifications. So there we're cleaving parts of the protein out, or we are adding units, adding amino acids to the end. We could be adding little caps or tails. There's all kinds of changes that can occur. But once, as we do that in the ER, eventually we're going to package that that peptide, that's that too, into a vesicle. The vesicle will be conveyed by some kind of a motor protein, well, it would be kinesin typically, which would be walking along a microtubule, and that will take it then to the Golgi apparatus, which I'll draw like that. And the Golgi apparatus, in the Golgi apparatus, you'll have additional post-translational modification that'll happen, additional folding and such that can occur. But eventually, then, the Golgi apparatus will read some of the tags that this peptide molecule has had placed on it, and those tags will be used to place it in an appropriate vesicle, which will ship it off to its eventual location. So if it's going to be a neurotransmitter, a peptide neurotransmitter, or if it's going to be a receptor, or a channel, or some other kind of membrane-bound protein, then it'll be packaged in a vesicle again, hooked onto a motor protein, which will walk along more microtubules and eventually get to the cell surface. So this would be the cell membrane out here. And you'd have exocytosis occur if we're releasing the peptide hormone. Or if it's a membrane-bound receptor, then you'd be attaching that to the membrane once it gets that surface. That's kind of like the series. So like that's a big chunk of chapter 2, all in kind of one image. This is pretty much what that inner life of the cell video was all about. But I think it's important that you understand all this, because these proteins that we're talking about manufacturing, those are doing all these jobs that we've been talking about in chapters 3, 4, 5, and 6, as far as receptors and enzymes that make neurotransmitters and other kinds of things. So I think that's what I would say. What else can we talk about? Yep.
If there's an aggregation of mitochondria in a particular location, what exactly is it?
What's that telling? I think it's indicating that there's a need for ATP, because that's what mitochondria do, is they generate ATP. So one example of a situation like this is we saw that there were aggregations of mitochondria in the axon terminal. And so that means that that area, because the only reason those mitochondria be there is because you need to generate ATP, so you have to power lots of processes in that area. And so you just put the little power plants right next to it. It does mean that that particular area would need oxygen in order for that to work, but I think the key thing there that you're finding is that the mitochondria are telling you that area needs lots of ATP, so you put the ATP factories right next to them. That's this question. Yep.
Could you explain the optogenesis again?
Yeah, so the idea, optogenetics? Yeah. So the idea there, do I have a figure? I don't remember what chapter that it was. Is that 4 where we talked about that? Yeah. The idea with optogenetics is that this is a new technique where basically they discovered some channels, I think in bacteria, that sounds right, that are responsive to light. And so the idea is that these are 2 that are shown in this image, that if you have this channel rhodopsin 2, this was discovered in some bacterium or some other organism, and the cool thing about it is that whenever you shine blue light at exactly 460 nanometers at it, it will then absorb that light and open a channel. It is sort of doubling as a channel. So it's kind of like it's kind of like a nicotinic receptor in a way, nicotinic acetylcholine receptor, because it's binding its ligand and then opening a channel. The difference is the ligand is kind of it's a ray of light or a photon of light that's causing it to open. This is a different one. This is halorhodopsin, which is a different kind of a channel. But again, the idea is that it absorbs a specific wavelength of light, and when it gets hit by that particular kind of photon, it will close in this case. And I think that's right. And then that will then cause a change in chloride conductance. So the cool thing about this is not so much just that these were discovered in some random organism, but they have isolated the genes for these particular structures. And then the technology has been developed where you can insert these genes into other organisms like a mouse, and then cause these channels to be expressed in certain mouse neurons. And once you've done that, now you've got this set of channels that you can literally just shine lights on. And there's a picture of this little mouse with a thing sticking out of its head, where they've got a little fiber optic cable. Here it is. They've got a little fiber optic cable that is sending light from a light source down the fiber optic cable, and it's shining on this little mouse's brain. And this mouse is a mouse where you've inserted these genes for the one particular kind of a channel. So this looks like a blue light, so this is probably that channel rhodopsin protein. And we can cause those channels to open whenever we want under this experimental apparatus. So this is really nice, because always before, if you wanted to cause a neuron to depolarize from like you just wanted to determine that neuron's going to depolarize now, you had to poke it with a glass electrode, which is a traumatic experience for the neuron at least. You had to then inject current. It was sort of an artificial situation. Now we have direct control over whether or not a channel opens. And so we can directly then study what an organism is doing with that particular neuron that you inserted those genes into and created that channel. So that's kind of the idea. Do you have a question or? Okay. Does that make sense? Any questions on that? Okay. Good. Well, wait. Is that on that? Yeah.
Can they for their neurons, can they during this development, can they put it towards just a certain type of neuron?
Yeah, I don't know how that works, but I think they do have some ability to direct it to be expressed in particular neurons, but I don't know exactly how they do that. So I can't answer that one. I mean, it might be all of them. It might just be, most neurons express it, but then you're really specific about where you shine the light. But I don't know. I've kind of gotten the impression they can restrict it, but I don't know much more than what I've told you, unfortunately. Gets in the news all the time, though. At least the last couple of years, I've seen a lot of stuff coming out just because it's kind of neat. But I think it's a really powerful approach that's going to open a lot of doors. Caitlin. Mm-hmm. Right. Right. Yeah. So we didn't really, so I guess there's two interpretations of size. I guess really we talked about the strength and then the length of a stimulus. I think those ones would be more where I would gear you towards. Size could mean like how large of an object, like how much space on a person's skin is being touched by an object. I won't get back to that. But yeah, the key thing about strength of a stimulus is basically just that we see, like this experiment showed us, that you see increased frequencies of action potentials the stronger the stimulus. So that's pretty much the story there. So it's action potential frequency, even though, in this case and in this case, these action potentials are all pretty much the same size here versus here. So the amplitude of the action potential does not matter. What matters is how many action potentials are occurring per unit time. And then like the length of the stimulus would basically just be how long of a period of time are those action potentials being conducted up the axon. So, here it's this constant stimulus, and so you have action potentials forever. If the stimulus only lasted for this period of time, you'd expect that very quickly, once the stimulus was removed, you'd cease to have action potential. So that might be length of stimulus. Size of stimulus, typically, at least the easy way to think about it is like a touch receptor or a pressure receptor. Those might be displayed or arrayed all around my hand. So you've got each different spot that I touch on my hand. I'm stimulating a slightly different pressure receptor. And I can tell in my brain where each one of those lives. And certainly the stimulus that I get when I press a large object against my hand is going to stimulate lots of those individual receptors simultaneously, which means many different axons going up to my brain will be stimulated simultaneously. Whereas if I had a point, I don't have I use my fingernail. If I just touched one little spot, that's only going to be stimulating one or maybe a few pressure receptors versus lots of them. So that might be more like the size. I think one of the key points, again, is just this idea that every action potential within an axon is pretty much the same as every other action potential within that same axon. So that doesn't vary. The amplitude of the axon never varies. It's an all or none phenomenon. The frequency certainly varies a lot. Yeah.
I just have a weird question related to that.
Yeah.
So when you visualize that in the graph, all of the action potentials we ever see just kind of go up and back down. Can they go up and stay up for a long time? Is that what you mean by the frequency?
No, I mean you have one after another after another. So they won't do that, and the reason they won't last is that in order for an action potential to the reason an action potential rises is because of those voltage-gated sodium channels. Those inactivate by the time you get up here, and the only way they will ever open again is if you allow them to reset by getting back down to threshold. So in fact, it is the case that the action potential anyway will never stay up here. It will have to return because there's just no upward stimulus once those sodium gates close, and that's due to this globular subunit. This guy. Now, there's a different thing, though, that can stay elevated. And that is that when you're looking at postsynaptic potentials, you know what? That was more of a homework question. And let's see. So, because postsynaptic potentials, when we're talking about the response of a postsynaptic cell to its input, those are not action potentials. These are going to eventually maybe cause an action potential to happen in the axon. But here what can happen, for example, is you might have a barrage of action potentials. So it's one action potential after another, arriving at the synaptic terminal. And each of those is a distinct, discrete entity. And each one will cause release of neurotransmitter in that spot. However, those neurotransmitter bursts will then move over and start to stimulate the postsynaptic cell, and those can start to overlap. So there you do see an overlap. That's temporal summation. But again, that's not driven. These are driven by ligand-gated or neurotransmitter-gated sodium or cation channels. Mostly they're sodium channels. They allow some potassium through too. They're not driven by the voltage-gated sodium channels, though. Yeah. Yeah, the IPSB. Yeah. Yep. No, that's fine. Thinking about the best way to answer the question. So here's what I'd say. What the Goldman equation, it's in chapter 3. What the Goldman equation allows us to do is for any given set of concentrations, darn it,